Have you ever wondered, when looking at a child, who his or her father is? Polymerase chain reaction can give you the solution you need to that problem.
Did you know that in 2015, the global PCR market was valued as approximately $2 billion? That's because PCR, polymerase chain reaction, is a revolutionary technique discovered by Nobel that won the Nobel Prize in 1993 due to its capability to rapidly undergo DNA replication. PCR amplifies DNA, allowing analysis of very small amounts of DNA or damaged DNA.
Without PCR, many of the techniques that will be discussed in this investigation would not be possible, such as paternity testing, forensic applications, and medical projects like the Human Genome Project.
Here are the materials needed. Taq polymerase is a heat-stable enzyme that catalyzes reaction. Because the chemical bond orientation of Taq polymerase allows for a high heat capacity in comparison to ordinary DNA polymerase, so that the bonds of Taq polymerase won't break or denature when heated. DNTPs are used to terminate the sequencing process because they lack the 3-OH. DNTPs allow the extension of DNA strand, so polymerase begins synthesizing new nucleotides from the N primer. And primers are short single strands of RNA or DNA, which are usually about 18 to 22 bases, that serves as a starting point for DNA synthesis. It is required for DNA replication because enzymes that catalyze the reaction, DNA polymerase, can only add new nucleotides to an existing strand of DNA. The thermocycler is the laboratory that is used to amplify segments of DNA. And the proper solution, which is necessary to create optimal conditions for activities of DNA polymerase, often contain trace hydrogen chloride, potassium chloride, and sometimes magnesium chloride. And the divalent cations, which are usually magnesium 2+, are positively charged, so in this reaction, they bind to the alpha phosphate group of DNTP and helps in removal of beta and gamma phosphate from DNTP.
The process of PCR takes place in the thermocycler, which is an instrument that effectively automates temperature cycling and incubation times. Prior to the introduction of the thermocycler, PCR was a lengthy process involving the transfer of samples between water baths of different temperatures and requiring much precision for timing each step perfectly. With the thermocycler, automation of PCR became a reality. The process of PCR has 3 steps, and it begins with denaturation. In denaturation, the double-stranded DNA unwinds into 2 separate single strands. The hydrogen bonds that hold the complementary bases of the DNA together are very strong, so it requires a lot of heat energy to break them up. So as a result, denaturation occurs at a very high temperature of 94 degrees Celsius. This process takes approximately 15 seconds, and it is crucial that the high temperature is maintained throughout the whole process. It is also very important to look at what exactly is going on at the molecular level of this step. As the temperature increases, the atoms move at higher speeds, and the vibrational frequency of the atoms increase. Now we have the second step of PCR, which is annealing. After denaturation, we are left with 2 single strands of DNA. They are complementary to each other, and they run in opposite directions. Annealing is when the DNA primers attach to the single strand of DNA. Annealing occurs at lower temperature, from 45 to 60 degrees Celsius. The lower temperature is necessary for the DNA primers to attach via hydrogen bonding. With a lower temperature, the partial match between the DNA primer and the template DNA will be stable enough, and you would get greater amplification in the whole process. Greater compatibility is needed at a higher temperature because otherwise the DNA primer and the template DNA will be broken apart from each other. Whereas at a lower temperature, the primer and the template do not need to be as compatible because they do not need to bind so tightly to prevent being separated by the heat of the reaction. The step of annealing takes approximately 10 to 30 seconds. Now we have the last stage of PCR, which is elongation. In this step, the temperature is increased to allow for the Taq polymerase enzyme to do ' job. Taq polymerase attaches to the DNA primer and adds the DNTPs randomly in the 5' to 3' direction. Elongation ends when there's no hydroxyl group left to react with the phosphate group on the DNA strand. The result is a 2 double-stranded sequence of target DNA, each containing an original strand and a newly synthesized strand. The duration of this step typically depends on the length of the DNA sequence, but it usually takes around a minute to copy approximately 1,000 DNA bases. These 3 stages are repeated 20 to 40 times, increasing the DNA copies each time. The new DNA strand that is generated can actually become the new template DNA for the next cycle of PCR. There are many copies of the primers and Taq polymerase floating around in the reaction in the thermocycler, which can then result in a larger amount of DNA molecules that can be synthesized through the whole process.
Now that we know how PCR or polymerase chain reaction functions, we can explore some of its many applications. One of the most significant applications of PCR involves the use of the gel electrophoresis apparatus. With so much DNA rapidly produced, PCR has allowed researchers to use gel electrophoresis more commonly as well. This chamber is able to separate various mixtures of deoxyribonucleic acid, or DNA, based upon the segment's different lengths by loading a gel with DNA and running an electrical current through the gel. The gel electrophoresis technique is based upon the fundamental need of the system to lower its potential energy and become more stable. DNA is negatively charged because of the phosphate group and its sugar phosphate backbone, and as such, it is loaded into the wells of the gel's negative terminal of the gel electrophoresis chamber. When the electrical current is applied, the DNA will then be attracted to the positive terminal of the chamber, because by traveling closer to the positive end, the negatively charged DNA will be reducing the distance and in turn make the potential energy more negative by the equation that states potential energy is proportional to the product of 2 charges over the distance. Gel electrophoresis differentiates DNA molecules by size because of molecular structure. As learned this year, when visualizing molecules, they should not be 2-dimensional, but rather 3-dimensional. Larger DNA fragments will be slowed down by the constitution of the agarose gel because it will have a harder time navigating the pores. Smaller fragments will be able to move easier, thus will travel farther and appear to travel faster in comparison to larger DNA fragments. Thus, PCR has paved the way for gel electrophoresis, which in turn has paved the way for many further applications. PCR has made many forensic applications possible because we can now concentrate on a single DNA segment of interest rather than an entire sample. The segment of interest can be rapidly amplified to make hundreds of copies in a short amount of time that can then be run on multiple gels, increasing efficacy of criminal investigations, for example. At a murder scene, DNA of blood at the scene can be isolated and copies can be made of the DNA fragments. Loaded onto a gel, the samples of different suspects' DNA may then be loaded in adjacent wells, and running the gel will produce different band patterns that can be compared for similarities as seen above. It is important to recognize that this image was taken under UV light, and this is because a fluorescent tag known as ethidium bromide was mixed into the agarose gel solution before it cooled. As we learned in chemistry, different molecules have different absorption maxima, and ethidium bromide absorbs wavelengths of 210 nanometers all the way to 285 nanometers, which corresponds to ultraviolet light or UV light.
Lithium bromide is excited by the UV light, and as such, it emits an orange light. An identical band pattern produces strong evidence for a suspect being guilty of the murder in a general electrophoresis chamber. The same logic may then be applied to paternity testing, where the DNA of the child can be run against the DNA of multiple potential fathers, and whoever has the greatest similarity in band pattern to the child is the father. However, it is important to realize that the band pattern will not be identical between father and son, because the child also receives DNA from his or her mother. Finally, following a natural disaster where a human is unrecognizable, PCR can be used to amplify the DNA found in the human's remains, and it can then be run on multiple gels to find a match to discern the deceased person's identity. This leads us into another field of applications where PCR has also paved the way for medical breakthroughs to become truly applicable when in 2003 researchers claimed they had successfully finished the Human Genome Project. The goal of this project was to successfully sequence the entire human genome, and PCR played no small part. By rapidly amplifying segments of interest, PCR sped up the process of mapping techniques used to help identify the sequences of the human genome. Now completed, the Human Genome Project has allowed for new fields of personalized medicine to take form. Nutrigenomics takes identified segments of interest from the Human Genome Project that are linked to one's particular conditions, and these are amplified by PCR to have many copies to analyze. Then, the best possible foods and nutrition to aid that person's condition are recommended. Disease subjectivity can also be discerned now by running one's DNA on a gel, because the band pattern can be matched to the normal band pattern of the human genome or a mutated pattern that may indicate disease, and this can reveal how prone an individual is to a specific disease. Rapidly amplified DNA from PCR can make cloning easier to identify because an identical band pattern will indicate 100% similar genetic material, which will indicate a clone and is irrefutable, and because of the volume of fragments produced from PCR, drug testing can be sped up and efficacy can be improved faster so that drugs can hit the market faster and save more lives. Without PCR, projects like the Human Genome Project would have taken forever to complete, and paternity testing would still be something of a luxury.
So in conclusion, PCR is an enzymatic process in which a specific region of DNA is replicated to yield many copies of a particular sequence. There are three major steps in PCR: denaturation, annealing, and elongation. The first step, denaturation, is when the double-stranded DNA unwinds into two separate single strands. The hydrogen bonds that hold the complementary bases together are very strong, so it requires a lot of heat energy to break them. As a result, denaturation occurs at a very high temperature. Annealing is when DNA primers attach to a single strand of DNA, which occurs at a lower temperature. And the last step, elongation. It occurs when tag polymerase attaches to the primer and adds the DNTPs randomly in the form of interaction. Elongation ends when there is no hydroxyl group left to react with the phosphate group on the DNA strand. PCR is a process that amplifies DNA, allowing analysis of very small amounts of DNA or damaged DNA samples. Therefore, the process has revolutionized DNA replication, and in turn has many applications medically and forensically.
Thanks to PCR, the Human Genome Project was able to be completed in a fashionable amount of time, and we can do things like find out who the murderer really was at a crime scene.